we have actin and myosin. Okay? Actin is the thin protein, and myosin is the thick. I try to remember these by actin, thin, and myosin is this big old M. You see the letter M, it's big and thick. So actin and myosin. Okay? Myosin, the protein filaments in that make up myosin look like a golf club. Okay, they look like a golf club. So, two golf clubs intertwined, no less. So we have our myosin head here, and we have our myosin tail. And a whole group of them are bunched together and held together here to make our myosin filament. Okay, our myosin filament. And they are specifically arranged, we'll learn today when we talk about the sliding filament, in this manner for muscle contraction. Now, our actin filament here, that's our red, that's noted here by the red dots, okay, the red beads being held together. That's actin. And you notice there's two other proteins associated with actin, and that is troponin, that looks like these three rocks being balanced together, okay? And the green rope or twine, tropomyosin. Now, if we look, let me blow this up here. If we look closely, here is the binding site for the myosin heads. And that tropomyosin is blocking those binding sites. So when tropomyosin is blocking the binding site for myosin, myosin cannot attach. And if it cannot attach, we cannot have muscle contraction. So this is where troponin comes into play, because as we see here, on our troponin, there's a calcium binding site. When calcium binds to troponin, it will cause a structural change, right? And with protein, a change in structure means a change in shape, in function. Yes. So that change will move the tropomyosin away from the myosin binding site. And now the myosin head can attach. Now this myosin head can attach to the actin. Okay, so tomatoes. Yes, on the vine. Yep. All right. So how's this all situated here to make our muscle contractions so effortlessly, and we don't even have to think about it every time we move a finger or even breathe. Okay? So again, this is the myofibril that has all the myofilaments. Okay? So if you notice, we've got dark, light, dark, light, dark, light. What did we call those in histology? Striations, yes. Okay? So here we have our sarcomere. Now remember when we talked about cells, and the cells are the basic unit of life. Okay? Can't get any smaller than a cell and still be a living organism. Okay? Our sarcomere is the basic unit of contraction for our muscles. Okay? We can't break down our muscles any smaller and still have muscle contraction. So this is the lowest we could go. And it's really cool how it is organized. So we have consistent muscle contraction. Okay? So a sarcomere is from one Z disc to the next Z disc. Okay? So you see that zigzag protein, that is the Z disc. Okay? So the Z disc is the zigzag, and that is going to define what a sarcomere is. Okay? We have our actin directly attached to the Z disc. And then we have our myosin in the middle. Okay? And that is connected to the Z disc by this little spring-like protein called connectin. And it is going to act like a spring. It keeps the myosin filament in between the actin filament. When we have contraction, those Z discs will come closer together. And when we relax, it bounces back. So it's just like the shocks on our cars. They bounce back into original place. Okay? So we have connectin, actin, myosin. Okay? Now within this sarcomere, we've got our M line, which is for middle, our middle line. Okay? We've got our H zone, and if you see our H zone only has myosin filament in it. Only myosin. Okay? There's no actin in the H zone. The A band here has actin and myosin in it. And then the I band only has actin and connectin in it. So, that A band, okay, that A band having both actin and myosin in it, that is the darker striation. That's the thick, dark striation. And that makes sense, right? Because we have myosin and actin in there. So we got a lot of protein, whereas this light band is the I band. Okay? So again, if we think I band, thin, actin, that makes up the I band. Okay? Whereas the A band, myosin, right? Myosin's a thick letter, big, thick, chunky letter. So is A when you compare it to I, right? A is a big, thick letter. Okay? Just hopefully that'll help you and not confuse you on the structure of a sarcomere and how to identify it. Okay?
What's the green thing again?
The green thing, that's the connectin. Connectin.
Is that another protein?
Yes. Yep. And that is strictly holding the myosin in place, keeping it in the middle. And I like that, you know, it's a double helix sort of protein, and it's springy, so it will bounce back. Okay. All right. So let's take a look at our list here. Let's zoom in. And let's focus. Okay. So figure 10.5 here has Z disc, M line, A band, I band, and H zone. We've talked about the myosin thick filaments, the tail and the head. You can cross off the ATPase. Okay, the myosin ATPase, just cross that off. that is another protein, and no models show this protein. We talked about the thin filaments with actin and then the tropomyosin, troponin with it, and the myosin binding site on the actin. This cross bridge cycling, cross all that out. That's physiology. That's, there's nothing I can show you. There's no anatomy, there's no what is this when it comes to this cross bridge cycle.
So that will be on the lecture exam.
Correct, that'll be on the lecture exam, that's more appropriate for the lecture exam. We talked about the myofibril and the fiber when we talked about the structure of it, the sarcolemma. Remember, that is the cell membrane of the muscle cell. It's the sarcolemma. Okay, what we didn't get to is the sarcoplasmic reticulum and the T-tubules. Okay? So here we have our muscle cell. Here we have our muscle cell.
Yes.
Thank you.
Okay, it's Tuesday morning.
There you go, it's Tuesday morning. Okay, here we have our muscle cell. And you'll notice there's this network here, okay? There's this green tube in between these blue networky things, netty looking things, okay? This dark blue network is the sarcoplasmic reticulum. The dark blue is the sarcoplasmic reticulum. Okay? And that acts just like the endoplasmic reticulum, except it's in the muscles, so we call it sarcoplasmic reticulum. Okay? And that green tube that's in between them, that is the T-tubule. And I think down here, we have a little bit, it's the sarcoplasmic reticulum, T-tubule, and you see this little tube going up? That is why we have holes in the sarcolemma, is that every one of these T-tubules opens up into the exterior of the muscle cell. And they are arranged this way, and they're arranged around each and every one of the myofibrils in this particular order. So when we have muscle contraction, we have the entire muscle contracting. And we have the entire muscle cell contracting.
That helps it to like move as a unit.
As a unit, yes. Yep. Perfect. Okay, so this blue netting, kind of like what you get that netting on like peaches, or some tomatoes, or roses, rosebuds can have some styrofoam, thin styrofoam coverings to help protect them. That's the sarcoplasmic reticulum. And then we have our T-tubule, and then our next sarcoplasmic reticulum. Okay? Now notice this is right over the sarcomere, and that's very important. In lecture, we'll talk about all this mess and go, ah!
Yeah.
Okay?
This part scares me.
Yeah, this part is the tough part. Yeah, that's the tough part.
What figure is this?
This is figure 10-3, okay? And this spot in 168 is our very first encounter with physiology, true physiology. Because we're going to dissect down everything that has to happen for us to have muscle contraction. And it's a good place to start with physiology, because one, it kind of opens your eyes, and it's like, oh my God, I never even realized all this has to happen. But hopefully it gets you excited about learning all the physiology coming up in 169. Because that is heavy physiology, okay? With digestion and respiration and everything. So that's a lot of physiology there. All right. So there's our T-tubules and the sarcoplasm, reticulum. We saw the nuclei, and the next thing we need to talk about, okay? We know skeletal muscle is voluntary. So we have to tell our muscle to contract, to move, so we can do X, Y, Z. So we are attached, linked into our nervous system. And this is figure 10.7, okay? How we're linked into the nervous system is through the good grief, neuromuscular junction, okay? The neuromuscular junction. So what we have here, kept thinking TMJ. I'm like, no. So what we have here, here's our muscle cell, our myofiber. Here are our myofilaments, okay? We have our motor neuron, our axon to our motor neuron coming in, okay? Motor neurons move things, just like the motors in our cars. So whenever our motor neuron is stimulated, something's going to move, okay? So our motor neuron, the axon comes down in close proximity, okay, to the muscle cell. It does not touch. It does not touch. This right here is the neuromuscular junction, because here's the nerve part, and here's the muscle part. And this is where they are in close proximity. They are so close that your hair thickness is thicker than this synaptic cleft. Okay, this space between the two of them is nanometers thick, wide. Okay, so let's take a closer look at this motor neuron junction here. And as we look, here's the axon terminal, and it has vesicles, synaptic vesicles, and inside that synaptic vesicle is a neuropeptide called acetylcholine. And when the electrical impulse comes down, we'll learn more about this in lecture, comes down, it will cause these synaptic vesicles to fuse with the membrane of the axon terminal, and it will exocytose the acetylcholine. So acetylcholine is exocytosed out of the axon, and then it swims across the synaptic cleft. I always think of the English Channel, like people swim in the English Channel to get to the other side. Okay, it'll swim across the synaptic cleft, and on the sarcolemma side, here's the sarcolemma, we have receptors for acetylcholine. And once it binds to its acetylcholine binds to its receptors, you see the movement of ions. And again, we'll learn that more about that in lecture today. Now notice there are only acetylcholine receptors around the axon terminal. Okay, there's no acetylcholine receptors over here. Okay? We call the place where the acetylcholine receptors are the motor end plate, because the acetylcholine is going to be released from the motor neuron attached to the receptors there, and there's where the acetylcholine's gonna have its effect on the muscle cell. Okay? So this is the motor end plate of the sarcolemma. Alright, so that is figure 10-7. And we have another figure here, 10-8. It does, again, putting everything together. Okay. Okay, so we hit the axon, the junction, and we related all of the endomysium, perimysium, tendon, and fascicle. Is there any questions about the terms for lab number 9? Okay, so we do have a model, okay, that can show some structures better than others, okay? All right, so this is the muscle cell itself, okay? Because here's the sarcolemma and here's the holes that go down to the T-tubules. And on this model, the T-tubules are in dark blue. And the sarcoplasmic reticulum is this beige netting here. But again, we got the myofibrils, the myofilaments here. And this whole thing is the myofiber. Here's our nuclei. Here's the motor neuron coming down to form the neuromuscular. And this would be the neuromuscular junction area here, and the synaptic cleft and everything, and motor end plate. Can't really see that very well on this model. We can see the A zone, A band for the myosin, the I band for the actin, and then along the backside, here's the sarcolemma, and this structure here would be the endomysium, covering the whole thing, the endomysium. Okay, so this is the sarcolemma with holes in it, poke, poke, poke. Okay, and right along with it is the endomysium on the outside. And that's what you can see on that model. Alright. Okay, so any questions?
Do you have any other models besides that?
No.
Nice.
And do not for the histology part. For the muscle parts, oh yeah, we got little muscle man and arms and legs. Muscle man. Yep. But the histology part now. Alright. If there's no other questions, I'll let you guys use the rest of this time the way you see best. Okay? Next